My name is Guadalupe Bolanos, and my presentation will be over the patch clamp method. To begin with, I will be giving a little bit of background information as to what the method is, what it's used for, and how it came to be. It was developed by cell physiologists named Erwin Muir and Bert Sackman during the late 1970s. Now, they used their technique to study single-channel currents across the membrane patch of a frog skeletal, and they were actually able to discover the function of ion channels in a cell, which led them to win the Nobel Prize in Physiology and Medicine in 1991. This method, the patch clamp, made it possible to register small electrical currents passing through a single ion channel. Now, there was a similar technique called the voltage clamp by Alan Hodgkin and Andrew Huxley, but that method wasn't able to measure currents passing through a single cell channel because it didn't have the resolution to do it. Now, this refined technique is now used to study excitable cells such as neurons, cardiomyocytes, and muscle fibers. And its principle is to isolate a patch of membrane electrically from the external solution and record the current flowing into the patch. Now, the general use is to answer various physiological questions, investigate ion channels in non-excitable and excitable cells, to use the method in medical research, specifically in diseases related to malfunctions in ion channels, and pharmacological research. Consists of a glass pipette containing electrolyte solution that is tightly sealed onto the cell membrane. Now, this tightly seal will isolate the membrane patch electrically, and a current will be pumped into the channels, and the current that flows through the pipette will be recorded by the electrode connected to a highly sensitive differential amplifier. Now, this figure here comes from a study done by Dr. O'Neill and Dr. Tung. And they wanted to see if the membrane conductance will increase if electropermeabilization depended on fast ionic channels such as sodium and calcium. They decided to have two different cells, one bathed in extracellular-like electrolyte, which was your control group, and the other bathed in extracellular-like electrolyte containing your calcium and sodium blocker. Now, a micro patch clamp method was used. A microelectrode was pressed against the cell surface, forming a tight interaction with the giga-ohm seal resistance between the cell and the rim of the glass. Then current flown through the membrane came from a DC voltage, and the current was recorded. Now, the figures show here that indeed sodium and calcium blockers had little to no effect on the conductance of the membrane, indicating that the ionic channels do not affect how fast the conductance response occurs. So here you see that there's only a slight difference. So even though they were present, the membrane conductance was not increased. And that's concluded that because the conductance response was so similar between the both, the membrane breakdown in amphibian cardiac cells don't involve the modulation of sodium and calcium ionic channels. Now, there were some drawbacks. They had to make sure that the membrane pipette seal did not dislodge from the cell, and that it was consistent, or else the recorded conductance would be measured incorrectly. And they also had no way of being 100% sure that the morphological condition of the membrane after isolation was not damaged and did not affect the results. Now, the good thing is that they were able to demonstrate electricity-induced membrane breakdown through the use of isolated cardiac cells. And they were able to explain that membrane conductance does change during electropermeabilization, which was their main goal. Now, the question I would like for you to answer is, given the 5 different patch clamp configurations, which of the 5 is the most advantageous and why? Thank you for watching, and I hope you learned a little bit about the patch clamp method.